Hello, Mr. Allman here, and this is your Allmancast on chromosomal abnormalities. A problem with a chromosome, since a chromosome is going to represent a very large percentage of an individual's DNA, tends to be fairly severe, in many cases lethal. Unlike regular mutations, any problem that occurs to a chromosome is likely to affect a very large number of genes. Now, there are two major ways in which a chromosomal abnormality can occur. One is a non-disjunction, and the other is a rearrangement. Non-disjunction is going to be an event in which chromosomes fail to separate during anaphase. It could happen in meiosis 1, when you have the homologous pairs fail to separate, or it could happen in anaphase of meiosis 2, when the sister chromatids fail to fully separate at the centromere at that time. In either case, you're going to end up with either an additional chromosome or missing a chromosome from the resulting gametes. This is something that's only going to affect an organism if that happens in a germline. Now, a germline is referring to a division within a cell that is going to give rise to a gamete. And remember that gametes are sperm in males and eggs in females. If this mistaken division happens in any other cell, it's only going to impact the resulting cells. Whereas if it happens in the germline, then every cell in that organism is going to be affected and have that either extra or missing chromosome. For that reason, problems with non-disjunction are really only going to be significant when they are occurring during the process of meiosis and not during the process of mitosis. Now, large-scale rearrangements are going to be somewhat similar to mutations that you're aware of that are happening to DNA sequences, but again, this is happening to the chromosome as a whole, and so every large-scale rearrangement may impact hundreds or even thousands of genes. Four types. One is deletion, this is going to be the case if a piece of the chromosome is removed, and then whatever genes were located in that particular sequence are then going to be lost. Duplication is going to be if there is a copying of a particular section of a chromosome, and so we see that that DNA is going to be increased, and so the total length of the chromosome increases. An inversion occurs when the particular sequence of DNA within a chromosome flips, and so instead of A, B, C, D, E, you see that the D flipped upside down and is going to be present next to the A, so we have A, D, C, B, E. So in this case, this may or may not have a big impact depending on whether or not there were any genes that are located on those break points where the chromosome was inverted. And finally, a translocation is when a segment of chromosome is moved from one chromosome to another, to a non-homologous chromosome. So in this case, you can see the A and the B getting switched together with the M, the N, and the O. It's always possible that breaks in DNA can occur, and when those breaks occur, there are proteins within the cell that are going to stitch the DNA back together. And so sometimes it's possible that if there are two breaks, that the wrong segment of DNA can get stitched back in the wrong spot. Now, these alterations have a very uneven effect. In some cases, there may be no impact, and in some cases, it might be quite severe. So the next part of the Prezi details some important examples. Whenever we have a trisomy or a monosomy situation happening to a single chromosome, that is usually going to result in an early miscarriage. So there's either going to be a failed implantation, or very early on in the stage of development of the embryo, the checkpoints within the embryo are going to recognize that mismatch of chromosomes, and those cells are going to die, and the pregnancy is going to be terminated. The exceptions here are the ones in which it is non-lethal, and the one that is most important for you to know is Down syndrome. Down syndrome is also known as trisomy 21. Tri meaning 3, and 21 meaning 3, meaning the 21st chromosome. So Down syndrome is going to be a case when there are 3 copies of that 21st chromosome. There is always a different characteristic facial appearance that you can see in someone with Down syndrome. There's going to be a varying other degrees of impacts on both physical and mental capabilities of that person, and there is, though, a large degree of variance between that. Now, trisomy 21 is not lethal because the 21st chromosome is quite small. As you can see, it appears down here at the bottom of this karyogram, and because it's a small chromosome, has only a small number of genes. And so there is ultimately less DNA impacted by having that extra chromosome. Some of the other common trisomy mutations or monosomy mutations occur on the sex chromosomes. In general, these are going to be tolerated. The one example is Turner syndrome, which is monosomy X. In this case, the person is going to be phenotypically female, so they will have female genitalia, but they will be sterile. This is associated with a lot of other physical problems, including heart defects, and so the lifespan and the quality of life is often going to be a little bit less than average for someone with Turner syndrome. Klinefelter's is a case when we have a trisomy on the 21st chromosome, so the X is doubled. The person is phenotypically male because of the presence of the Y chromosome triggers the development of the penis and the testes prior to birth. However, there is reduced fertility or actual infertility, and it is possible to see some female secondary sex characteristics like overdeveloped breast tissue in comparison to what you would see in someone who has just an XY chromosome. Multiple X is not really a problem. So I should say multiple Y and multiple X chromosomes are not really a problem. And if you think about the reason why, first of all, with the Y chromosome, it has such a small number of genes. You can see the Y chromosome pictured here next to the X. And because it has few genes, if there are extra copies of that particular chromosome, those genes aren't being used much, and so it doesn't cause much confusion within the cells. The major function of the genes there is to increase production of testosterone and direct the formation of the testes, and so once birth has occurred, there's even less importance for that Y chromosome. Multiple X is also not a problem because even though the X chromosome is very big and has a lot of genes in it that are vital for life, you only need one copy of it to be able to function. And what happens in females who normally have two X chromosomes is something called X inactivation. So that extra X chromosome is going to be deactivated early on in development, and so that only one of those X chromosomes is actually used for transcription and translation. So if you have two X's and one is inactivated, then the same system, if you have 3 or 4 X's, will inactivate those additional X chromosomes. And so for this reason, a person with multiple X chromosomes is not going to be affected in any way. Finally, let's take a look at what the effects are of some of the other types of chromosomal abnormalities. If you have additional sets of chromosomes to creating something like a triploid or a tetraploid, in animals that's not going to be tolerated. That's lethal, and so that's going to result in an early miscarriage. In plants, the plants have other ways of being able to deal with those extra chromosomes, and so that's not going to be lethal in the case of most plants. When there are chromosomal rearrangements, like a translocation, a deletion, or an insertion, sometimes those are detrimental and sometimes those are silent, meaning that they have no impact. And so it really depends on where on the chromosome and how much of that chromosome is actually affected. So there are a couple examples on here showing some of the different known cases of translocations and the impact that that has when it does have a noticeable impact on phenotype. So that is the Almond Cast on chromosomal abnormalities. As always, please let me know if you have any questions.